As we study prokaryotes, we do want to understand what are their unifying characteristics, and then also what are the ways that we do classify these types of organisms. So first thing is, what are prokaryotes? So what is it about them that groups them together? Well, at the most basic level, these are organisms that are composed of prokaryotic cells. So we do have to understand, what are prokaryotic cells? What are the features of that cell type? These are going to be basically cells that have no membranous compartmentalization of the cell's interior. So the inside of the cell is not going to have separate membranes that divide it up into different isolated areas. This essentially translates into the fact that there will be no membrane-bound organelles. So we will not have things like a nucleus, a chloroplast, mitochondria, vacuoles. All of those are not going to be present because those would be membrane-bound organelles. If we were to talk about the actual genetic information and the chromosome of the prokaryotes is made of DNA, but in this case it's a circular double helix of DNA, and it's not going to be associated with histone proteins. Another thing to understand is, again, there's no membrane compartments, so this is going to be in the nucleoid rather than in the nucleus. We will not have a nucleus when we're talking about the prokaryotic cells. So that's the type of cells that they're actually going to be made of. Now, if we talk about some other general characteristics that we do have with prokaryotic cells, well, first of all, these do tend to be organisms that are unicellular. So as unicellular, if we just write that right here, that means that they do tend to be organisms that are made up of just one cell each. There are some exceptions to that, but that is for the most part going to be what we tend to see. And these prokaryotic cells, they do exist in three basic shapes. We have coccus, bacillus, and spiral. And if we identify those in the picture here, the coccus ones are going to be the circular ones or the spherical ones here. These are not always going to be perfectly spherical. They can be more oval shaped. They could be more kind of kidney shaped. The bacillus ones are going to be the ones that are more rod shaped. And then the spiral ones, you've probably figured out which ones they are. They're the ones that have more of this type of arrangement. So there's long and slender, but then they do tend to spiral around. Those are the general shapes that we do have, or the basic shapes that we do have, but there are some variations on that, and you can see some of those variations in this image. So coccus would be spherical, but then we can have some that are kind of an intermediate between the coccus and the bacillus. So we call those coccobacillus. You can see the vibrio down there, which are basically rod shaped, but then it's a bent rod. And we can have spirillum. Spirillum is going to be a rigid spiral bacteria. And then we can have the spirochetes, which is going to be more of a flexible spiral bacteria. The pleomorphic that you see down there is basically variable. So these pleomorphic prokaryotes are going to be ones that vary in their overall shape and size. And we will have certain genuses of the prokaryotes that do tend to have pleomorphic shapes. Another characteristic of prokaryotic cells is that many of those organisms are capable of forming endospores. Endospores are very stable, durable, resting stages. We refer to them as spores. These are basically a defensive strategy that the cells will use. And it's a defensive strategy when conditions get really rough, so under hostile or unfavorable conditions. And so these can withstand extreme temperatures many times. They can withstand extreme dehydration. So what this means for us as humans is that these can be things that are really hard to kill off or eliminate when we are, say, sterilizing food products. So just heating food sometimes is not going to be able to get rid of those endospores. So it is important for the food industry because many of the prokaryotes, they can form these. And if they do survive in that particular food product, then they can later return to their normal state, and they can cause a variety of human illnesses. And anthrax and tetanus are just a couple of these serious diseases that they can cause. It is important to understand that these are not reproductive structures. If it was a reproductive structure, we would be taking one cell and we would be producing new cells from that, and we're not doing that at all. We have one cell, and that one cell is kind of bunkering down to survive some harsh conditions, and it does then produce the endospore. New endospores can be produced in 8 to 10 hours. So that's relatively quickly that you can have a cell that goes into this just really durable kind of lockdown state. If we do talk about the production of new cells, the production of new cells does occur all asexually. So when we talk about asexual reproduction, this is going to be clonal reproduction. And what we mean by clonal reproduction is that we are not going to be getting genetic variety in this case. So genetically, those new cells that are produced are going to be identical to each other. The primary way that this asexual reproduction is going to take place in prokaryotes is going to be with the process of binary fission. If we look at the binary fission process here in this image, the first thing that has to happen is the DNA has to be replicated. So that single circular chromosome that we have is going to be copied so that we have essentially 2 circular chromosomes. And then those 2 circular chromosomes are going to move apart and separate from each other. Once they have moved apart and separated, we will start to see the forming of a new cell wall down the center of this elongated cell. And eventually it will form completely, and these cells will become separate from each other. And we do have what we then call our 2 daughter cells. Because it is asexual clonal reproduction, those 2 daughter cells will be genetically identical. Again, they're the same as each other. They're also the same as the original cell that they did come from. We do sometimes have a special type of binary fission, and we call this snapping division. And the reason we call it snapping division is because the way that the cell wall is going to be formed down the middle is actually going to result in this kind of snapping process when they do separate. With the snapping process, there is going to be a thickening of the inner cell wall, and that thickening of that inner cell wall is going to put tension on the older wall. And eventually what this does is it does cause an actual snapping process. Other types of division, first of all, we have some microbes that actually retain their own identity while they give rise to offspring. So rather than have one cell that basically goes away when we produce the new cells from it, which is what we get with binary fission, you have one cell that stays there, and it is kind of the parent, and it kind of gives rise to these new spores. That's what we see in this image right here. That's really similar to what we have when we're talking about fungi. If we look at other types of reproduction, we do have what's called fragmentation. We can have a budding process as well. The budding process is what this image is showing us. With budding, we're going to, first of all, have to have some copying of the DNA. And then once we have copying of the DNA, we'll see an overall elongation and kind of budding out, separating out of one part of the cell, and it's going to essentially pinch off, and we will have our new daughter cell, and you'll have the original parent cell, which is going to be larger, that is still there. So you have the parent cell, and then you have this young daughter cell that has separated off. All of these different reproductive strategies, they do result in genetically identical cells because, again, it is asexual reproduction that is taking place here. There can be different arrangements to the prokaryotic cells, and these different arrangements that we get, these are based on 2 key aspects of that binary fission. And what this is the plane of cell division, and that's basically whether they divide this way, or maybe it's a diagonal division. And then also whether those daughter cells do remain attached to each other or whether they do ultimately separate from each other. We see the most variation when we're looking at the spherical cells, so the coccus cells. If you look here, we can have just 2 cells that are attached together, or we can actually get long chains of cells attached together. In some cases, we can get 4 cells that remain attached, and we call that a tetrad. And then again, if we have other variations, we can get clumps of cells. And those clumps of cells, they can have a cube-like shape to them. They can have just an overall kind of irregular arrangement that makes kind of a blob of cells, for lack of a better description. Different types of prokaryotic cells will have different characteristic arrangements to their cells. The bacillus cells, they can come in different versions or arrangements as well. They can come in just two of them attached to each other. We can get a chain of them attached to each other. We don't have quite the same variety here, but we can have rows of them, and that is what we call palisade. And then sometimes they kind of stay attached to each other and they form kind of this V-shaped structure, and we just refer to that as V-shape. Again, which shape, which arrangement we actually have is going to be characteristic of specific types of prokaryotic cells, or what we have here are really specific types of bacteria. If we move on and talk about how we do classify the prokaryotes, so how we put them into different groups. Well, first of all, taxonomists, they have taken all of the living organisms and they have divided them into three main domains. Those three domains that they've divided them into are bacteria, archaea, and then eukarya. And when we're talking about prokaryotes, prokaryotes are these two categories right here. So these are going to be the focus that we're going to be looking at here, because both of these are made up of prokaryotic cells. And there is a bit of variety with the prokaryotic cells. So the main way that they will be classified is based on their rRNA sequences. So the ribosomal RNA sequences, so comparisons between them, and then the ones that are the most similar to each other will be grouped and classified together. Bergey's Manual of Systematic Bacteriology is the most accepted classification system that we do use, and that's the one that we're going to go through here. With that particular classification system, there are 26 different phyla that the prokaryotic organisms are going to be divided into. 24 of those phyla are going to be for the bacteria domain, and then 2 of those phyla are going to be for the archaea domain. So here is an overview of those different phyla that we're talking about. Notice that the archaea is down on the bottom. There are a lot fewer identified and characterized archaea cells compared to the bacteria. So we're going to go through the archaea phyla and their general characteristics first, and then we'll take a look at the bacteria ones. If we look at archaea first, archaea are prokaryotic cells. So they do have that characteristic of not having a cell that is subdivided into individual membrane-bound compartments. These archaea, one thing that is going to separate them from the bacteria is that these are prokaryotes that don't have true peptidoglycan in their cell walls. So they have cell walls, but they can be made of different materials. They're also going to have a difference in the overall arrangement of their membrane lipids. Their lipids are going to have branched or ring-form hydrocarbon chains, whereas the bacterial cells will have more linear or chain-like unbranched hydrocarbons. And then another thing about the archaea is that the initial amino acid that they do have in their polypeptide chains is going to be methionine. So basically, when the ribosome goes through the process of translation, the first amino acid that it will always be incorporating into the new protein that it is making is going to be methionine. This, again, is different from what we have in bacterial cells. So all three of these characteristics that we've listed here, they are characteristics that are very specific to archaea, and they are not the things that we would see in the bacteria group. So when we get done just kind of separating out the archaea can be grouped into 2 main phyla. Those are the phyla that you see here. Now, archaea, if we talk about their reproduction, they do typically reproduce by binary fission. They can also reproduce by other ways. So they can reproduce by budding. They can reproduce by fragmentation. Both of these would be asexual reproduction strategies. So these would produce genetically identical cells. They can have different shapes to them. They can be cocci, they can be bacilli, they can be spiral, they can be pleomorphic. So in other words, they can be spherical, they can be rod shapes, they can be spirals, they can have a variety of shapes, is what we're saying there. And we do have the variety of shapes, or the pleomorphic, in this image right here. Notice it's just an overall very odd shape to it. That's characteristic of pleomorphic organisms. And then if we talk about their cell walls, we said their cell walls are not going to be made up of peptidoglycan. So that is not an archaea thing. But they do have cell walls, and those cell walls are going to be made of a variety of compounds. These are unique compounds or unique cell wall arrangements for Archaea. And these are going to include proteins, glycoproteins, we'll have lipoproteins, and then also some polysaccharides that do make up that overall Archaea cell wall. Now, some Archaea are extremophiles. And that's one of the things that Archaea are kind of known for, is the fact that they are extremophiles. And what that means is that you do have some members of this group that live in really extreme environments. So by extreme environments, these are going to be living and surviving and thriving really in environments that other typical organisms are not going to be able to survive in. So that could be at very extreme temperatures. It could be at extreme I'll say oxygen levels. It could be at extreme salt levels. So if we look at just a couple examples, one group of Archaea is what we call the thermophiles. Thermo has to do with temperature. And so for these are prokaryotes whose DNA, RNA, cytoplasmic membranes, their proteins as well, they don't actually function properly if the temperature is below 45 degrees. And most organisms need to be below 45 degrees Celsius in order to survive. So that's a pretty warm temperature that we're talking about with 45 degrees. So this is not all Archaea, and there are some bacteria that will fall into this group as well. But we do have a bunch of Archaea that fall into this group. We tend to find them in places like hot springs. They thrive in those conditions. There are actually some thermophiles that can't survive below 80 degrees Celsius. So they have to have even hotter temperatures in order to survive. Again, the DNA, RNA membranes, they're going to have a different general arrangement to them so that they can actually survive in those types of conditions. We have this particular one, Geogemma, and this one can survive 2 hours at 130 degrees Celsius. So that's just to show you how unusual these are. We call them extremophiles because they live and they survive in very extreme conditions. 100 degrees Celsius would be boiling. So this is 130, which is significantly hotter than boiling temperatures. And this one can survive 2 hours at that temperature. Now, the benefit for some of these is that because they can survive at such high temperatures, they have enzymes inside of them that are designed to work at really high temperatures. And that has been used to the advantage of researchers because those enzymes can be used for recombinant DNA technology. In the lab, many times reactions need to be performed at very high temperatures, and here we have enzymes that are ready to work at those temperatures. So a lot of those enzymes are isolated and used for cloning and recombinant DNA purposes. If we look at this picture here, we can see a lot of red to orange pigments, and that is because there is a great abundance of halophiles that grow in these solar evaporation beds, which we're seeing here. These are located near San Francisco, and again, they're very salty, so the organisms that tend to survive there are halophiles. One of the most studied halophiles is Halobacterium. The name is a little misleading because it's not a bacteria, it's an Archaea. This particular one, it does survive in really salty environments, and this one has the capability to perform photosynthesis. But one of the main differences with this and other typical photosynthetic organisms is that this one collects light with a special purple protein instead of a pigment. And this special purple protein that it uses to collect light is what we call bacteriorhodopsin. Another type of extremophile is the methanogens, and we do have a number of Archaea that do fall into this group. The methanogens are going to be obligate anaerobes, and that means that they are not able to survive in the presence of atmospheric oxygen. And one of the reasons why they're not able to survive in the presence of atmospheric oxygen is that they will not have the necessary enzymes and methods to detoxify toxic oxygen compounds. What these methanogens are going to do is they are going to take carbon dioxide, hydrogen gas, other organic acids, and they will be converting them into methane, which is an organic molecule. So they will, in the environment, be converting organic waste into methane, and the environments where you do tend to see these growing are going to be anaerobic environments, so bog type environments in many cases, and they will generate that methane gas.